This pneumatically activated microfluidic compression device can be used to study mechanobiology in 3D hydrogel cultures of growth plate chondrocytes.
The microfluidic device is cost and time effective because it can simultaneously generate multiple magnitudes of compression on small volumes of samples, and various microscopic imagings can be applied with the device.
Our method can be also applied to the study of mechanobiology of other cell types.
To set up the microchannel layer, first mix PDMS with a weight ratio of 10 prepolymers to 1 curing agent for 5 minutes. Next, place the mold on a foam pad and pour the mixture into the master mold. Degas the PDMS in a vacuum chamber for 30 minutes and sandwich the degassed PDMS with a piece of transparency film. Clamp the sandwiched assembly with a glass plate, foam pads, and plexiglass plates. Cure the PDMS at 80 degrees Celsius for 6 hours, and isolate the PDMS layer and the transparency film from the sandwiched structure. Activate the surfaces of the PDMS and a clean glass plate with a plasma cleaner for 1 minute, before bonding the PDMS onto the glass plate in the 80 degrees Celsius oven for 30 minutes. Then remove the transparency film. For preparation of the thin PDMS membrane, spin coat uncured PDMS on a transparency film at 1000 rotations per minute.